morning, everyone. I am Jeph Macaraig, and I am here to present our study titled Environmental Factors that Influence the Occurrence of Waterborne Pathogens in the Seven Lakes of San Pablo, Philippines. The Seven Lakes of San Pablo provide ecosystem services to the surrounding communities such as irrigation and water supply, aquaculture and fishing, and recreation and ecotourism. It also serves as area of commerce with several business establishments in the vicinities of the lakes, and the surrounding areas also provide location for residences and households. With the apparent increase in the anthropogenic activities in the lakes, regular monitoring and assessment should be conducted for conservation and to prevent further deterioration. However, regular monitoring of water quality in the Philippines includes several physico-chemical parameters, and fecal coliforms are the only biological agents monitored by authorities. Alarmingly, this group of bacteria may not be the only disease-causing organisms that might be lurking in these freshwater ecosystems. Of equal importance are parasitic protozoans that have been identified as one of the leading etiological agents of waterborne diarrheal outbreaks. Earlier studies have revealed that approximately 1.8 billion of the global population are exposed to fecal contamination through drinking water, resulting to diarrheal diseases, which accounts for the second major cause of death in children. Hence, the Project 3 of the Seven Labs research program focused on determining the extent of contamination of emerging waterborne parasitic protozoans in the Seven Lakes. Specifically, it aimed to detect the presence of Cryptosporidium, Giardia, Blastocystis, Acanthamoeba in the surface waters of the lakes, study the spatial distribution of the parasites for tracking of point and non-point sources, and to determine the correlation between parasite contamination and water physicochemical parameters. This study was conducted in the Seven Lakes of San Pablo City, Laguna. Water samples were collected in stations assigned in each lake representing the various lake and land uses in the area such as aquaculture, residential, agricultural, and commercial establishments. Physicochemical parameters were also measured. Water samples were filtered and the collected sediments were subjected to standard parasitological techniques such as immunofluorescence assay and in vitro cultivation protocols. PCR assays were also conducted for species, genotype, and subtype identification. Data were analyzed using the quantum GIS for mapping and distribution analysis. Correlation analysis was done to determine the relationship between parasite contamination and water physicochemical parameters. Our findings have revealed that the Seven Lakes are contaminated with the four target pathogens with highest and lowest contamination incidences recorded for Cryptosporidium and Giardia respectively. This difference in contamination incidence can be attributed to the higher number of Cryptosporidium oocysts that are shed in feces of an infected animal or human hosts, as compared to Giardia cysts. Moreover, Cryptosporidium has resistance to various disinfectants, has no seasonality, and may persist in the environment, which may contribute to higher chance of recovery or detection. Blastocystis was found in all the lakes. This parasite is characterized by having a cosmopolitan distribution, and due to its low host specificity, many animal species may harbor infection and serve as reservoirs that may contribute to environmental contamination. On the other hand, Acanthamoeba is ubiquitous in nature and can be found in both terrestrial and aquatic ecosystems. This graph shows the distribution of the parasites in the Seven Lakes. Results show that at least two parasites were found contaminating the lakes. Two parasites were detected in Pandin Lake, while all parasites were detected in Calibato Lake. Meanwhile, the lowest incidence of parasite contamination was recorded in Mohicap Lake. The differences in contamination levels may be linked to various factors and characteristics unique to each lake and the extent of anthropogenic activities in the lake's vicinities. Distribution analysis shows that contamination was common in areas where human habitations and animals were present. In Sampaloc Lake, 3 of the 4 parasites were detected in areas where residential and commercial establishments and animals were present. Sampaloc Lake is located at the city proper, where a high number of households and commercial establishments can be found. The lake also serves as an attraction frequented by locals and tourists and is the largest among the 7 lakes of San Pablo. In Bunot Lake, parasites were detected in areas where fish pens are located, Some areas of the lake near households that have livestock and companion animals were also contaminated with parasites. In Mohicap Lake, Cryptosporidium, Giardia, and Blastocystis were detected near the areas intended for recreational and aquaculture purposes. Mohicap Lake has the least number of households in its vicinity, and the aquaculture and recreational activities are relatively less compared to other lakes. While this may limit development and livelihood opportunities for the residents living around its premises, this results to less anthropogenic activities that translates to the low parasite contamination levels as reported in this study. All parasites were detected in Calibato Lake, specifically in areas near fish cages, households, and river outlets. Like the Bunot Lake, Calibato Lake has high aquaculture operations, and more than 60% of the lake area are occupied by fish cages at the time of collection. In Palakpakin Lake, all sampling stations were found to be contaminated with parasites. Parasites were detected near households, fish cages, and river inlet and outlet. Residents in the vicinity of Palakpakin Lake are also engaged in aquaculture for their income and livelihood. In Pandin Lake, parasite contamination was found highest near households where livestock animals such as pigs, ducks, and chickens are raised. Some were also detected in areas intended for ecotourism activities. In Yambo Lake, parasites were detected in stations where fish cages and some animals like horses and ducks were seen. Acanthamoeba and Cryptosporidium were also found in areas intended for ecotourism activities. Correlation analysis between the presence of Cryptosporidium and physicochemical parameters of the lake's surface waters revealed significant correlations between the presence of Cryptosporidium and total dissolved solids and water transparency. These findings highlight the effects of natural phenomena such as precipitation on the lake water quality. Changes in lake's physicochemical characteristics were possibly due to the runoffs brought by the high amount of rainfall during the collection period. Since human settlements and livestock and companion animals are evident in the vicinities of the lakes, the runoffs may not only contain nutrients but also some fecal matter contaminated with parasites from the surrounding communities. The isolated parasites from the surface waters were subjected to molecular assays for characterization and identification of possible sources. Cryptosporidium can be exploited as sentinel of fecal pollution and is a good candidate for source tracking as it exhibits host specificity. In our study, a specific region in the small subunit ribosomal RNA gene of the organism was amplified by nested PCR and sequenced. Our findings revealed that among the samples successfully sequenced, the dominant species contaminating Calibato and Yambo lakes are Cryptosporidium galli, baileyii, and andersonii, commonly infecting animals including cattle and poultry such as chickens and ducks. Among the three species identified, only Cryptosporidium andersonii has records of zoonosis as revealed in previous studies. On the other hand, Blastocystis is an emerging protozoan parasite with cosmopolitan distribution and is commonly detected in humans and animal species. Zoonotic transmission through water sources is possible because of its low host specificity. Results showed that subtypes 1, 2, and 4 were present in the lakes. Previous studies on Blastocystis have recorded the presence of these subtypes in animals such as rodents, dogs, and pigs, and are also zoonotic. These are some photos documenting the presence of households, some poultry coops, and other companion and livestock animals such as dogs, cows, and horses near the lake's shoreline. These factors are the potential sources of parasite contamination in the lakes. Protozoan parasites such as Cryptosporidium, Giardia, and Blastocystis enter the aquatic ecosystems as they are passed in the feces of infected animal and human hosts. On the other hand, Acanthamoeba is an opportunistic free-living amoeba found in various natural and man-made aquatic ecosystems that causes cutaneous lesions and sinus infections, keratitis that may lead to vision impairment, and a rare but fatal encephalitis. Although a free-living organism, previous studies suggest that abundance of Acanthamoeba may be influenced by the presence of other bacterial contaminants in the environment, including E. coli and other coliform bacteria, as these free-living protozoans feed on these bacterial cells. Moreover, more recent studies have revealed that Acanthamoeba may be a potential reservoir of other microorganisms, including those with public health importance. This framework shows the transport of pathogens from sources or hosts to water resources. Here we can see that humans and animals both contribute to parasite contamination through excreta. Natural phenomena such as precipitation may also play an important role in the transport of pathogens from upland areas to water bodies through runoffs. In addition, stream beds can also be important repositories of pathogens that may be resuspended in the water column during storm events and natural upwelling or overturn. To summarize, the anthropogenic activities have increased in the past recent years due to the rich resources and livelihood opportunities in the seven lakes of San Pablo. However, despite the increase in the various activities in the lakes, water quality monitoring efforts are limited and other equally important parameters are neglected. This study provides evidence of contamination of emerging waterborne protozoan parasites such as Cryptosporidium, Giardia, Blastocystis, and Acanthamoeba, and contributes to the scarce data on waterborne protozoan parasite contamination in Philippine freshwater ecosystems. Possible sources of contamination include the presence of animals in households near the lake's shoreline. The presence of these parasites in the lakes may serve as a tool to identify point and non-point sources of fecal contamination, as these are usually shed in feces of both humans and animals. The ongoing works of the project include molecular analysis such as genotyping and subtyping of isolated parasites for further characterization and host source tracking, data analysis for the risk factors assessment on water, sanitation, and hygiene practices, biosecurity, and environmental factors, and communities' knowledge, attitude, and practices toward lake contamination and disease transmission. And we also are doing collaboration with local government units and other agencies to revisit lake and land use plans and management of the lakes, and possible inclusion of waterborne protozoans in regular water quality monitoring efforts. We would like to thank the DOST Grants and Aid, National Research Council of the Philippines, and other partner agencies for their help in the successful implementation of the Seven Lamps program. This project is led by Dr. Rachel V. Vipalier, and we would also like to acknowledge the Parasitology Research Laboratory of IBS and the graduate students who worked under this project. Thank you very much for your attention.